Hi students, and welcome back to my channel Bonding with Chemistry. I am Dr. Seema Singh, and in today's video I will explain you the topic indicators from the chapter Acids, Bases and Salts for class 10. Before we get into the details of what are indicators and the types of indicators, it is important that we understand why exactly do we need indicators when we are dealing with acids and bases. Let me explain this with an example. Suppose I give you a tomato and a bitter gourd and ask you to identify which of the two is acidic or basic in nature. You would say that tomato is acidic in nature. Why? Because it is sour in taste. Or bitter gourd is basic in nature because it is bitter in taste. But what if unknown chemicals are provided to you in the laboratory? Would you risk to taste it? No. It is not advisable to taste any chemicals in the laboratory, not even if it is sodium chloride, your common salt, because these chemicals may be contaminated or they could be harmful. They could also be toxic, irritant, corrosive, flammable, or all of these in nature. Then, how do we find out the nature of a substance? Don't worry, you just have to remember a tagline that chemicals are to be tested and not tasted, and the substances that help in testing them are known as acid-base indicators or simply indicators, which are defined as dyes or mixture of dyes which help us in identifying whether a given substance is acidic or basic in nature. Now, acid-base indicators are broadly classified into four categories: natural indicators, synthetic, olfactory, and universal indicators. Natural, synthetic, and universal indicators are also your visual indicators because they help us in identifying acidic or basic solution by showing a change in color. We also have olfactory indicators which help us to identify an acid or a base by producing a change in odor or smell. Now, an advantage of such indicators, that means olfactory indicators, is that they ensure the participation of visually impaired or color blind students in laboratories. Isn't that great? In my current video, I'll explain in details natural indicators. Thereafter, in my next video, I'll take up synthetic and olfactory indicators. Later on, in a different video, I'll take up universal indicator, which forms the most important indicator, as you might have both theoretical as well as practical based question from this topic. The speciality of universal indicator is that it not only tells you the nature of a substance, meaning whether a given substance is acidic in nature, neutral, or basic in nature, but it also tells the strength of the substance. That means whether a given acid or base is weak in nature, strong in nature, very strong in nature is also indicated by universal indicator, an example being pH paper. Therefore, it becomes the most important indicator from this chapter. Let us begin with the first type of indicators, that is your natural indicators. As you can see by the name itself, natural, that means these indicators are derived from natural sources, that is nature. For example, your turmeric, your litmus, you have your red cabbage, beetroot, flowers, maybe hydrangea, your petunia. All these are natural indicators and I'll take them one by one in this video. Let us begin with the most common example of natural indicator, our very own turmeric, also called as Haldi. Now, Haldi is a very common spice that is used in cooking. Also, it is one of the easiest available acid-base indicator because it finds space in every kitchen and has been there since ages. You must be familiar with Haldi stains. Some or the other time, you must have stained your clothes with Haldi. In the picture, you can see the Haldi stained shots of my toddler. He got this stain while attempting to eat his lunch all by himself in school. Big boy. I just thought of illustrating how turmeric can be used as an acid-base indicator with the help of a simple activity of removing the stain. The moment I dip this cloth in detergent solution, you would get the same observation if you try soap. The stain turned reddish brown in color, and when I washed it with excess water, the stain turned yellow again. Obviously, it was lighter in shade. Now you know that soaps and detergents are basic in nature. You can always go back to my introductory video if in case you missed that part. So we see that the activity illustrates the turmeric changes from yellow to reddish brown in basic medium. Then what happens in acidic medium? If you add turmeric to an acidic solution, for example, while preparing potato vegetable, after a while you add acidic tomato puree and thereafter turmeric, you will see that it retains its color and the vegetable turns yellow in color. Well, if you're still wondering what happened to the turmeric stain on my child's cloth, don't worry, I was able to get rid of it on repeated washing. Trust me, it is a tedious job. If you don't believe me, try this activity at home, but do it with a rough handkerchief. So we can see that in neutral solution and acidic solution, turmeric is yellow in color, whereas in basic solution it is reddish brown. So you need to remember only this color change. In neutral and acidic solution, your turmeric would be yellow in color, whereas in a basic solution it would turn reddish brown. In fact, you can also use turmeric paper, also known as curcuma paper, because the scientific name of your turmeric is Curcuma longa. So this paper you might find in your school laboratory. In case you are not able to locate it, you can prepare this indicator by a simple technique. You can take filter paper. You must have seen filter paper or the blotting paper. Prepare turmeric solution, dip your filter paper into the turmeric solution and let it dry. So it's nothing but a paper saturated in turmeric solution and allowed to dry. In neutral solution and acidic solution, just like your turmeric, the paper would remain yellow in color. Whereas in a basic solution, the paper would turn to reddish brown. Moving on to the most widely used indicator in the laboratory, the litmus indicator. The word litmus has been derived from an old word that means to dye or color. It is nothing but a water-soluble mixture of various dyes that have been extracted from lichens. You're going to study about them in biology, or you must have already studied. Basically, lichens are nothing but composite organisms made up of algae and fungi. Now, litmus is one of the oldest forms of acid-base indicators. Now, litmus can exist in the form of solution. Generally, the solution are red litmus solution and the blue litmus solution. You can see in the picture here. This is your blue litmus solution and this is your red litmus solution. Or it can also be adsorbed on filter paper, just like your curcuma paper. And you can produce litmus paper. Again, they are generally red litmus paper and blue. So you can have red litmus paper, red litmus solution, or blue litmus paper, blue litmus solution. These are all your litmus indicators. Remember that neutral litmus solution, the original litmus solution, is purple in color. In acids, this purple color would turn red, and in bases, your purple color would turn blue. So you need to remember that your natural litmus indicator is nothing but purple in color. In acids, it turns from purple to red, whereas in bases, it turns from purple to blue. My dear students, litmus indicator is the most important topic from this entire video. Litmus test would be part of your various practical activities, not only in class 10, but higher classes if you opt for science stream. It will also form the basis of quite a few reasoning questions in your board examination. So, how do we perform this test? The litmus test is performed by placing a small drop of sample onto the colored litmus paper. You will have, say, red litmus paper or blue litmus paper. You're going to put a small amount of sample with the help of a dropper onto the litmus paper and notice or observe the color change. In case you have litmus solution, then you're supposed to pour few drops of litmus solution with the help of a dropper into the given substance and observe the color change. Now, acid would turn blue litmus red whereas it will have no effect on the red litmus. You can see it has no effect on the red litmus, whereas it has turned blue litmus red. Whereas bases would do just the opposite. They would turn red litmus blue and it will have no effect on blue litmus, as you can see in the picture here. My dear learners, it is very important that you remember the color change produced by acids and bases on the corresponding litmus. You can use the following shortcuts to remember this. The first one being acid, the last alphabet is D, red, the last alphabet is D. So acids are going to turn red. Now what are they going to turn red? Obviously blue litmus. The bases, the first alphabet is B and blue, the first alphabet is B. So bases are going to turn blue. What are they going to turn blue? Red litmus. Or you can remember the other shortcut that I have created. B, R, B. Yes, one of the most widely used acronym in online chatting. Be right back. Look, B for basis turns red litmus blue. So if you remember this, that basis turns red litmus blue, acids would do just the opposite. Simple. Let us take up a recent question from NCERT to strengthen our knowledge about the litmus test and the color change associated with it. The question says, you have been provided with 3 test tubes. Let's say 1, 2 and 3. One of them contains distilled water. Let us say the first one contains distilled water and the other 2 contain an acidic solution and a basic solution. If you are given only red litmus paper, so you have just one red litmus paper, how will you identify the contents of each test tube? Just remember the acronym that I have just taught you, BRB. Bases are going to turn red litmus blue, whereas acids will have no effect on the red litmus paper, and your neutral solution will also have no effect on the litmus paper, whether it is red or blue. So what you're going to do is, we're going to dip red litmus paper into the solution. Since we have just one paper, either you can dip the strip into the solution or you can divide the paper into 3 and then add a drop of distilled water, acid and base respectively on the paper and then perform. Totally your discretion. So we have a red litmus paper. I dip it into the first test tube. I see that there is no color change. In the second test tube, I see there is no color change. Whereas when I dip it into the third test tube, I see that the red litmus paper has turned into blue, which shows that the third test tube contains a basic solution. Now remember that you have been provided with just one red litmus paper. Now you have this red litmus paper. It is now converted into a blue litmus paper. This part has converted into a blue litmus paper. Now third one has been identified. So we leave this test tube. We have 1 and 2 to be identified. Now, if I'm going to dip blue litmus paper into acid, so if base turns red litmus blue, your acids are going to turn blue litmus red, right? So I dip this strip into first solution and I find no change. Whereas when I dip this strip into the second solution, I see the strip has turned into red, which indicates that the second test tube contains an acidic solution and therefore the first one contains distilled water. Let us move on to the next topic. Yes, my dear learners, you can prepare your own natural acid-base indicators by using various colored vegetables, fruits and flowers. Remember, you should take the colored part of the plant, not white. For example, red cabbage, beetroot, blueberries, hydrangea, petunia, geranium. Now these are flowers, all these. You can take this interesting activity as a project for science exhibitions in school. It would be fun demonstrating the various color changes by adding just a single type of indicator in different solutions. For junior students, it would be just magical. Let me explain this through a couple of examples. Students, please know that I would be taking up only NCERT examples, but in detail for better understanding. That means I'll explain you red cabbage activity, beetroot, hydrangea, petunia and geranium. Let's begin with red cabbage. Red cabbage is also known as purple cabbage because of its color, or red crot as its leaves are, you can see, dark red or purple in color. You can prepare the red cabbage juice by following the process of grating, blending and straining, and the juice would be your acid-base indicator. Now, this juice is going to give you a variety of colors depending on the strength of the solution. That means how acidic or basic a given solution is. The juice of the red cabbage is red, pink or magenta in acidic solution, whereas in neutral solution it is purple in color. In basic solution, it gives a variety of colors, as I said, depending on the strength of the solution. As the solution becomes more and more basic, you can see the color change from blue to green to yellow. So what you can remember is that in acidic solution, red cabbage is red in color. In neutral solution, it is purple in color, that is its original color, whereas in alkaline solution, it is greenish yellow in color. Let us take up a reasoning question based on the above. On cooking, red cabbage will normally turn blue. What would you add to retain its red color? So if you have to retain its red color, you need to add some acid because red cabbage in acidic solution is red, pink or magenta in color. So you can add vinegar or any acidic fruit in the cooking pot. Now acidic fruit could be your lime, it could be your tomato, and your red color would be retained. Another interesting example of homemade acid-base indicator is beetroot, your chukandar. In fact, there's a group activity in NCERT which asks you to prepare your own indicator and then you are supposed to note down the various observations. The chukandar extract or juice can be prepared in a similar fashion just like your red cabbage, and then you can use it for determining whether a given solution is acidic, basic or neutral in nature. The beetroot extract, that means the original extract, is deep red in color. You can look in the picture here. In acidic medium, the color is still red. That means it retains its color in acidic medium. In neutral medium also it will be deep red. Whereas in basic medium, the color is purple. And if you have a highly basic medium, a strong base, the color would be yellow. What you are supposed to remember is that in acidic solution, it is red. Neutral solution, it is red. Whereas in basic medium, it is purple. You need not remember highly basic medium. This much is not required. Coming on to flowers as natural indicators. The beautiful flowers of Hydrangea have different colored petals depending on the nature, that is pH of the soil. You can see in acidic medium it would be blue in color, in neutral soil medium it would be purple in color, in basic medium it would be pink in color. So the petals of Hydrangea, especially a species Hydrangea macrophylla, can be used as an acid-base indicator. In neutral solution, the color is purple. In acidic solution, the color would turn into blue, whereas in basic solution, the color would be pink. Another beautiful flower that can be used as acid-base indicator is Petunia. It exists in a variety of colors. There are different colors. They are even white, but you cannot use white color petals for preparing acid-base indicator. It does not have that pigment or the dye which would change color in acidic or basic solution. So, the extract from its colored petals can be used as acid-base indicator. In acidic solution, the color would turn to reddish purple, whereas in basic solution, the color would be violet. I'm sure you can now answer this multiple choice question. A bud of Petunia became reddish purple after first shower of rain. What does it indicate? And the answers, you have 4 options. First shower causes buds to blossom, first shower of acid rain, first shower causes buds beautify, or all of these. Do post your answer in the comment section below. The last natural indicator that I am going to take up in this video is geranium flower. It is a beautiful flower with 5 petals and it exists in variety of colors like white, pink, purple or blue. But dear learners, please note you cannot use white petals as acid-base indicators. My dear learners, do write back in the comment section how many of you really counted the number of petals in the picture. Coming back to the topic, in acidic solution the color is orange-red. You can see in the picture over here, whereas in basic solution the color is blue. I am sure by watching this video you must be well versed with the topic of natural indicators. I have summarized the entire content of this video in this table, which would help you to recall and remember the various natural indicators and their respective color change in acidic, neutral and basic medium. Dear students, please note that the indicators are arranged in the order of importance for your board examination. the first two being the most important. I would suggest you to pause the video and note down this table and thereafter memorize it. But remember, you should be well versed with the color change by red litmus and blue litmus. With this useful table, I come to an end of this video. In my next video, I will take up synthetic and olfactory indicators. So, if you haven't subscribed to my channel yet, please do it now. Also, if you liked my video, do give it a thumbs up. And for latest update on my videos, hit the bell icon. Thanks for watching and stay bonded, my dear learners.